Hello friends, welcome back to our channel, The Exploring Medicò. So last time we had discussed about antigen and our today's topic is antibody. So let us initiate our today's topic antibody by discussing of its basic definition. So it is a large Y-shaped protein molecule produced mainly by plasma cells to neutralize pathogens like bacteria and viruses. It is also known as immunoglobulin, present as Ig. So it is presented as Ig in short form. Antibodies are present in serum, tissue fluid and mucosal surfaces. So antibodies are produced by B lymphocytes as we can see in this diagram. So now let us discuss immunoglobulins in details. So basic structure of immunoglobulins are, it is composed of 4 polypeptide chains, 2 identical light and 2 identical heavy chains. It is linked by disulfide bonds. The light chain is similar in all the immunoglobulins. The light chain occur in 2 varieties, that is kappa and lambda. The light and heavy chains are subdivided into variable and constant regions. And each heavy and light chain contains amino terminal in variable region and carboxy terminal in constant region. Now let us discuss classification of immunoglobulins. So based on its structure and antigenic nature of H chain, the immunoglobulins are classified into 5 main classes, that is immunoglobulin G, gamma, immunoglobulin A, that is alpha, immunoglobulin M, that is mu, immunoglobulin D, that is delta, and immunoglobulin E, that is epsilon. Now it's time to discuss immunoglobulin G in details. So it is the most abundant class of immunoglobulin in serum. It constitutes about 80% of total immunoglobulin. IgG, that is gamma, present in blood, plasma and tissue fluids. In serum, it exists as monomer. It contains less carbohydrate than other immunoglobulin. Immunoglobulin G has half-life of 23 days, the longest of all the immunoglobulin isotypes. So IgG is longest of all the types of isotypes. IgG crosses placenta and provides natural immunity to the fetus and neonate at the birth. It acts against bacteria and viruses by opsonization. It neutralizes toxins. IgG activates complement by classical pathway. Subclasses of immunoglobulin G. So IgG1, IgG2, IgG3 and IgG4. So biological function of these subclasses are Ig1, 3 and 4 crosses the placenta and protect the fetus, and Ig3 activates complement. Now Ig1 and 3 together binds to Fc receptor on phagocytic cells, monocytic and macrophages and mediate opsonization. Now let us discuss immunoglobulin A, that is alpha, in detail. It constitutes about 10 to 15% of total immunoglobulins. As we discussed, IgG constitutes about 80% and IgA constitutes about 10 to 15%. Now IgA is present in milk, saliva, tears, mucus of respiratory tract, digestive tract and genitourinary tract. In serum, it exists as monomer. In external secretions, it exists as dimer called secretory immunoglobulin, secretory IgA. It has J chain and secretory piece, and its half-life is 6 to 8 days. As a dimer, so there is a 2 molecule and secretory component, and there is a J chain. Functions of immunoglobulin A. IgA, it provides local immunity. Secretory IgA binds to surface antigens of microorganisms and prevents its attachment and invasion of the mucosal surface of respiratory and digestive tract, that is immune elimination. Secretory IgA provides important line of defense against Salmonella, Vibrio cholera, and gonorrhea, influenza virus, and poliovirus. Secretory IgA present in breast milk, and it protects newborn during first month of life. It activates complement by the alternative pathway. IgA promotes phagocytosis and intracellular killing of microorganisms. Immunoglobulin M. So, it accounts for 5 to 10% of total serum proteins. It is polymer of 5 monomeric units, therefore it's pentamer. Held together by disulfide bond and J chain. Molecular weight of immunoglobulin M is 9 lakh to 10 lakh. It is a millionaire molecule. Half-life is 5 days. As the name suggests, pentamer, so there are 5 molecules. So this is basic structure of pentamers of IgM. So there is a disulfide bond, J chain, and 5 molecules. So most of IgM present intravascularly. Most of that consists of 80%. It is present in low concentration in intracellular tissue fluids. It cannot cross placenta, as we had discussed in immunoglobulin G, that it crosses the placenta and provides immunity. And this one IgM cannot crosses the placenta. Okay. The presence of IgM antibody in serum of newborn indicates congenital infection. The earliest immunoglobulin to be synthesized by fetus in 20 weeks only. First immunoglobulin to be produced in primary response to antigen. Relatively short-lived, hence its demonstration in the serum indicates recent infection. Functions of IgM. It agglutinates bacteria. Activates complement by classical pathway. It causes opsonization, believed to be responsible for protection against blood invasion by microorganisms. Immunoglobulin D, that is immunoglobulin delta. This structure is similar to IgG. It constitutes about 0.2% of total immunoglobulins. The molecular weight of IgD is 1,80,000. Half-life is about 3 days. It exists as monomer. So IgD together with IgM is major membrane bound immunoglobulin on unstimulated B lymphocytes and acts as recognition receptor for antigens. Now let us discuss immunoglobulin E in detail. So it is a structure similar to IgG. It has 4 constant region domains. Molecular weight of IgA is 1,90,000 and its half-life is 2 days. It exists as monomer. It is heat-labile, inactivated at 56 degrees Celsius in 1 hour only. Mostly present extracellularly and it does not cross placenta. It produced in the lining of respiratory and intestinal tract. It is known as regine antibody. It does not activate complement or not activate agglutinate antigens. It mediates immediate hypersensitivity reaction and responsible for symptoms of anaphylactic shock, hay fever and asthma. It plays a role in immunity against helmintic parasites. Now, dynamics of antibody production. So, there are two types, primary immune response and secondary immune response. So, primary immune response is there is a latent period, gradual rise in antibody production taking days to weeks, plateau reached, then antibody level declines and antibodies are IgM. So we can plot the diagram as I had shown in this figure. Now secondary response. So second exposure to same antigen. Memory cells are a beautiful thing. Recognition of antigen is immediate. Results in immediate production of protective antibody, mainly IgG, but may seen in some IgM. So we can plot both together in graph form as I had described in this figure. Like gradually it increases. There is a primary and secondary part. So you can also plot the diagram in this form. Now antibody production. So initial antibody production of IgM lasts 10 to 15 days, followed by production of IgG. It lasts 4 to 5 days. Without continued antigenic challenge, antibody levels drop off, although IgG may continue to be produced. So thank you friends for listening me, and if you like my video, please subscribe our channel, and if you have any doubt, let me know in my comment box. Thank you. Stay updated with our channel.